These are the presenters of the course. As you can see, today we have Mathieu and Nynke, and on Tuesday Cyril was also entertaining us. And we have the support from Geraldine and also from Hugo Costa, who is doing all the technical support, which is very needed, in my case at least. So we are talking about this paper, published in 2016, which had a flow diagram in it, describing a number of steps that could be taken for the risk assessment of food components and products, making use of, as much as possible, use of the NAMS approaches. And as I stated on Tuesday, which differs in many aspects of the presently prevailing methods. This is just to make sure that you are very afraid of going into this, because this is the complicated scheme that we are going to explain to you in more detail. So don't be afraid. This is the complicated system, but we go through it in a proper way. Well, on Tuesday we had introductions on the general overview on how hazard and risk assessment is done at the moment and how we could see it in the future. And we also had some more details on the cytotoxicity of it. To remind you, we had also a poll. We asked the participants where they came from industry, from academia or elsewhere. This was about, well, more equally presented, which is nice. We also had people from asked the people whether they were familiar with toxicology, risk assessment, and cell cultures, and there were quite some people that were, but also that weren't. We had about 40 people in the meeting. Today we had a registration of 88. I'm not sure how many are now in the call, but normally that is always a little bit less. So the next presentation will be given by Mathieu Vinken on adverse outcome pathways. And then we have the presentation by Nynke on physiologically based kinetics. We will have then a presentation by Nynke also on the qui vive, on the quantitative in vitro in vivo extrapolation. And finally, I will give an introduction to the hands-on training in July. So without further ado, I would like to ask Mathieu Vinken.
Yeah, thank you everybody for attending, and thank you also Ilse Europe and especially Bas, of course, for organizing this. I think this is a very relevant and especially a timely topic. NAMs all over the place, I would say. And I would say one of the cornerstones of these NAMs are mechanisms. And you might know that in the field of toxicology, mechanisms, well, it's not conceptually new, but it has gained increasing attention, especially over the past few years, in particular under the form of so-called AOPs, adverse outcome pathways. So in this presentation, I would like to give you some background on what AOPs, adverse outcome pathways, actually are, but also how you can use them in practice. So for purposes in the field of both toxicology as such, but especially risk assessment, because in the end, this is what we want to do. So I hope you see also my next screen now saying AOPs. Again, AOPs have been there now since 2010, but especially I want to emphasize again that these are not conceptually new, because that was already known before, of course. People like to give very fancy definitions to AOPs. This is one of those. I think it was one of the very first definitions that has been introduced by the OECD, saying that an AOP is a conceptual construct that portrays existing knowledge concerning the linkage between a so-called direct molecular initiating event, an MIE, and an adverse outcome at a biological level that is relevant to risk assessment. This is a very long definition, but basically in practice, what it comes down to is that an AOP is exactly what you're looking at the screen. It is a, let's say, a schematic representation of the mechanisms that underlie specific types of toxicity. So that typically stretches from the molecular level, and it may go all up to the population level, as you can appreciate from this slide. It does not necessarily have to. Typically, population level AOPs are usually ecotoxicological AOPs. For the more human toxicology AOPs, it might end, and it usually ends at the organism level or even at the organ level. Now, in each AOPs, and there are a lot of out there, I will be coming back to this later on, there are at least two basic elements. So the first type of elements are these green boxes, as you can see here, which are the key events. And as the name already suggests, these events are really critical for the AOP to take place. And a second requirement for a key event is that they should be quantifiable. So somehow you should be able to measure them. Also important is that there are two special cases of these key events, namely at the starting point of an AOP, which is called the molecular initiating event, so the MIE. So that would be the interaction of, for example, a chemical, but it can be a stressor in general, so it does not necessarily have to be a chemical. So an interaction of that stressor with a biological system. So that's the first special case of a key event. And then the second special case is at the other end, that would be the AO, the adverse outcome. So that would be the apical endpoint, if you will. So that's the first type of elements that are critical for an AOP, the key events. And then the second aspect of an AOP would be the links between the key event relationships, which are shown here by these arrows. So they can be direct or indirect, and I will be showing you some examples on this later on in the presentation, but in the most recent OECD nomenclature, this is called adjacent and non-adjacent kind of relationships, if you will. What this figure also tells you is that different types of information can be fed into an AOP, so in chemical and in silico, so computational kind of data, if you will, in vitro data. In vivo data that is available, so animal data, but also epidemiological and clinical data, so human data, if that were to be available. So these AOPs have been introduced in 2010. They mainly originate from the field of ecotoxicology, but then they have to become applied also to the field of, let's say, human toxicology. Now, a couple of years ago, these principles, AOP principles, have been introduced. You can see them here. There are 5 of them. So the first one says that an AOP is not chemical specific. So very often I hear that people construct an AOP for a chemical. No, that's not correct. The idea is that any chemical or any stressor can trigger an MIE in an AOP. And that's not the chemical, but rather the chemical interaction at the MIE that is specific. So if you want to build what people call a chemical AOP specific one, or that usually is not an AOP, but it's what we call a mode of action. So that's really different. So the second principle says that AOPs are modular. The rationale here is that AOPs should be flexible, clear, and transparent to use. And that's very important here. So we always make the comparison between these key events and these Lego blocks that you still might know from your childhood, or yeah, maybe some people still play with them right now. But the idea behind this is that you can actually use these Lego blocks and thus these key events in any kind of AOP, and that's really important. The third principle says that an individual AOP is merely a pragmatic unit for development and evaluation. Yeah, indeed, in the early days of AOP, so the AOPs that were, introduced at that time only had one starting point, so one MIE and one endpoint, an AO. But of course, we know that toxicity in reality is much more complex than that, because you can have parallel pathways, you have crossing of pathways. And that is exactly why we have these so-called AOP networks, which are the real functional units of prediction. So these can be used for real-life applications, and I will be showing you some examples further on. And among the many applications is, for example, they also would enable you to, let's say, kind of capture different interactions in AOPs, and also to, let's say, at least model the effects of chemical mixtures, so rather than individual compounds. And then the very last, but certainly not the least, AOP principle is that an AOP is a living document. So it's a dynamic document that should be continuously updated by feeding in new data, but it should be fit for purpose. And that's really important. And I will be kind of showing or demonstrating this principle to you at the end of the presentation. Now, basically, everybody can come up with a new AOP. So you should have, of course, some background knowledge, but it's not that this is really rocket science, if you will. If you have expertise in a particular field and you know where to find the data, then you should be able to actually come up with an AOP. And for that particular reason, the OECD, which has been and still is pretty instrumental in the field of AOPs, came up with a system to assess, to evaluate, if you will, newly postulated AOPs. And to do this, we rely on what we call the tailored Bradford-Hill criteria. Some of you might be familiar with the Bradford-Hill criteria. That's something that comes from the field of epidemiology, I think from the 1960s or so, which basically try to establish the minimum criteria in order to establish a link between events. And you know, with the start of AOPs, or with the introduction of AOPs in 2010, these existing Bradford-Hill criteria have been somehow a bit customized or modified in order to really fit the purpose of AOP, so hence the tailored Bradford-Hill criteria. I will not go through this in detail because that would be a presentation or even a workshop on its own. But just in a nutshell, what is it all about? Well, if you assess, evaluate a newly postulated AOP, then you should basically answer a bunch of questions that relate to the biological plausibility of the key event relationships, the essentiality of the key events, and especially the empirical support of the key events and the key event relationships. And this may seem very trivial, but I can assure you I did this kind of exercise myself for one of the AOPs back in 2013. And if you want to do this according to the OECD guidelines, it's really a lot of work. Fortunately, these days, we have things like artificial intelligence that can help you to facilitate or to make it at least a bit more easy. So, well, actually, you should consider throughout the tailored Bradford-Hill criteria assessment the different evidence in a weight of evidence kind of approach. So depending on the quantity, but also the quality, of course, of the evidence and the data that you have available, the confidence in one or more of these criteria can be strong, moderate, low. So I already mentioned the OECD, and indeed they are very active. They still are very active in the OECD, in the AOP field. And one of the very nice initiatives already initiated several years ago, together with a number of other agencies like the US EPA, was the establishment of this AOP knowledge base, which is publicly accessible and available. For now, the most important module of this AOP knowledge base is the latter one, that would be the AOP wiki. I checked just right before this workshop or this webinar today, and as we speak, there are approximately 500 different AOPs that have been included in the AOP wiki. And they really, you know, relate to very different types of toxicity. So not only restricted to human toxicology, but also ecotoxicology. So if that were to be of interest to you, and especially I would encourage all of you to at least have a look, please go to the website. So again, it's very user-friendly. It's very easy to have a look at that. And even if you were to be interested, and if that were to be of interest to you, you can maybe even include your own AOP of interest. All right, in the remainder of the presentation, I would like to demonstrate some aspects of AOPs and their use. And I would like to do this by focusing on AOPs that relate to liver toxicity. And why is that? Because I think we all know that the liver is a critical organ in terms of toxicity because of a number of reasons. It's also a main target of toxicity. So just a bit of a recap. I assume that most of you know what the liver looks like, but anyhow, so this would be what you would see if you would look through a microscope and you would look at, in this case, a human liver tissue. So have a look at what you see on the slide on the left-hand side. In the upper part, you would see several 10,000s of this hexagonal structure, which is called the liver lobule, which is about 1 or 2 micrometer in diameter, and in which cells are organized radially around branches of a central vein. And at each of its 6 corners, a liver lobule meets 2 other lobules in an area which is here magnified below, called the portal triad, and that consists of branches of the hepatic artery, the portal vein, and also the bile ducts. Now, as is the case with all of the organs and tissues that we have in our body, also the liver harbors different cell types, and this is what is shown to you here on the right-hand side below. So the most important liver cells, so both in number, but also in, let's say, function, are the parenchymal cells, which are shown here in brown and which are called the hepatocytes. And they really can be considered as the workhorses, if you will, of the liver, because as we will be seeing further on, they do take care of most of what we call the liver-specific functions. But nevertheless, there are also some very important non-parenchymal cell types present in the liver, and this is also shown to you here in this figure. So for example, these cells here, the endothelial cells, that make up the lining of the blood flow in the liver that we call the sinusoid. And by doing so, they create the space which is called the space of this. And in the space of this, other cell types, non-parenchymal cell types occur. So for example, these stellate cells that take care of the storage of vitamin A in the body, and that, as we'll see further on, in case of liver toxicity, especially fibrotic kind of liver disease, adopt a very, very different kind of phenotype, and they also start to lose their vitamin A content. And then finally, in the sinusoidal lumen, there are a couple of non-parenchymal cell types present that take care of the immunological function of the liver, which is still very underestimated, I must say, especially in the field of toxicology, but nevertheless very important for things like idiosyncratic reactions. So for example, these Kupffer cells and these dendritic cells. Well, as already mentioned, the liver is a crucial organ. You cannot live without a liver, so it's really vital. And it does take care of, for example, the metabolism of lipids, carbohydrates, also the synthesis of essential proteins. Think about albumin, but also blood coagulation factors. This is all being taken care of by the liver. As already exemplified, also some nutrients and vitamin, like vitamin A, as already mentioned, can be stored in the liver. But certainly from the toxicological point of view, the most prominent liver function would be the biotransformation of foreign chemicals, which we collectively designate as these xenobiotics, as you probably know. And as already mentioned, as a consequence of this very important function, the liver also represents a primary target of systemic toxicity, so which can be manifested over the shorter term, so that would be days or a couple of weeks, but especially over the longer term, so months and even years. Think about alcohol abuse, for example. It might take a couple of years for your liver in order to, show some toxic manifestations rather. So indeed, different liver injury types are actually known, which can be characterized by different, let's say, clinical chemistry parameters, but also different histopathological patterns, and this is what you're looking at. But what they all share, what they all have in common, is a certain degree of cell death and inflammation. So for example, liver fibrosis, which as such is a liver scarring formation process that is the result of accumulation of extracellular matrix, and I will be coming back to this when I will be showing you the AOP. Then cholestasis, which is my own field of interest, which is a result of accumulation of bile, and that can occur inside of the liver, and that is what we call intrahepatic cholestasis, or it can occur outside of the liver, extrahepatic cholestasis. Please keep this terminology in mind because it will come back later on. Undoubtedly, the most studied type of liver toxicity would be the accumulation of lipids, is what we call steatosis. Actually, 1 in 3 Europeans suffers from liver steatosis, usually diet-induced. And at the histopathological level, at least two different types can be distinguished. And this is also what you see on this slide. So on the left-hand side, you have microvesicular steatosis. So you cannot even see the lipid droplets, but they are so small that they actually do not really affect, let's say, the localization of other cellular organelles. And that's really different if you look at the right-hand side, and that would be macrovesicular steatosis, in which the lipid droplets are very big in that respect, that they actually start to affect the localization of other cellular organelles like mitochondria, the nuclei, and so on. And finally, if not treated, then chronic liver disease can result in the onset of liver cancer. There are different types, but let's say the most prevalent one would be hepatocellular carcinoma. So given the fact that the liver is such a primary target and a frequent target of toxicity, it's probably also not a surprise to you that there are a lot of AOPs out there that relate to liver toxicity. And again, this morning I checked the AOP wiki, which I already mentioned, and there are about 30 to 40 different AOPs in the AOP wiki that specifically focus on liver toxicity. So I summarized, or I listed a couple of them on this, but also on the next slide. But the message for now is that there is at least one AOP out there in the AOP wiki that mechanistically describes one of the different types of liver toxicity that we shortly discussed on the previous slide. So acute liver injury, there are two for liver fibrosis. There is only still one for cholestasis, and I'm proud of this because this is the work of my own team in Brussels. Again, not surprisingly here is that there are a lot of AOPs that relate to liver steatosis, and as I will be demonstrating later on, most of them have now been merged in what we call an AOP network, and then also, of course, for the different liver cancer cell types, there are quite a few AOPs out there. It's not my intention, I really don't have the time to go through all of these different AOPs. I selected a couple of them for you just to illustrate the different aspects of AOPs as we already introduced at the start of the presentation. So that would be an AOP on liver fibrosis, officially called an AOP from protein alkylation leading to liver fibrosis. And just for your information, this was the very first AOP that has been fully endorsed by the OECD. So this means that it went through the entire, very thorough, I must say, process of evaluation of the OECD, including evaluation by 70 researchers. So you can imagine what kind of rebuttal that must have been. And it also has been published, actually, and I got the privilege to be part of this work. That was work that was led by the Joint Research Center of the European Commission. So it may seem very trivial if you look at the AOP, but I can assure you that there is a lot of work that is hidden behind all of these boxes and these arrows. So it starts with protein alkylation and covalent protein binding in the blue box, that would be the MIE, if you will. This then leads to hepatocyte injury, for instance, by apoptosis, leading to the activation of Kupffer cells. So remember that these are one of the immune cell types in the liver. This is then responsible for the expression of transforming growth factor beta-1, which underlies what I would say the most important key event in this AOP, namely the activation of stellate cells. So remember that under normal circumstances, these stellate cells, they store vitamin A. In fibrosis, they lose vitamin A, and they adopt a kind of fibroblast-like phenotype, which is associated with the secretion of a lot of extracellular matrix proteins or components. And this is exactly the very source of this scar formation. So this is what we call fibrosis. Also have a look at the different ways in which, by which these key events are linked with each other. So we are talking about these key event relationships now. And it is exactly this what takes a lot of work, because you have to show also by, assessing by means of these Taylor-Bradford-Hill criteria that you have enough evidence to show that they actually are linked, and also that this is the right sequence in which all of these key events occur. The second example I would like to discuss with you is an AOP network. So it's not a single AOP, it's an AOP network on liver steatosis, which, as you can see on the slide, actually results from merging, in this case, 10 individual AOPs that each have their own MIE, their own start point. And as you can tell here, the MIEs typically are, like nuclear receptors, activation, these kind of things. I will not go through all of them in detail. So what happens then is quite some transcriptional changes that relate to lipid metabolism. And the next step would be de novo fatty acids synthesis that goes hand in hand also with the increase of fatty acid influx from peripheral tissues. What happens next is a very important event, of course, a key event. That would be the accumulation of triglycerides in the hepatocytes, which you can observe microscopically. So it can be macro or microvesicular steatosis. And depending on this, you also would notice, this, let's say, distortion of other cellular organelles. So we tend to call this a fatty liver, but of course, I would say the more scientific, correct terminology here would be steatosis. So that's an example of an AOP network. And then the last example I would like to share with you is an AOP network on cholestasis that my own group in 2013 has introduced. So it was introduced at that time in compliance with the OECD guidance. And one of the main guidelines there was like, you need to focus on a single MIE. But for something that complex as cholestasis, we already decided to introduce an AOP network, although the concept of AOP networks was not, really known at that time. So that's why you have different MIEs on the left-hand side, sorry. So what you can see in the blue boxes. But for the sake of still complying with OECD guidance, we still decided to at least focus on one of the MIEs, which is shown in the dark blue box called inhibition of BSAP. And I apologize because I should have included here a list of abbreviations. Some of you might know that BSAP stands for the bile salt export pump. So that's one of the many transporters that takes care in the liver of the transportation, not only of bile acids, but also of a number of pharmaceutical drugs. And of course, if you inhibit this, then the first thing that will happen, and that would be then a key event, would be the accumulation of the bile acids. What happens next is something that is really specific to cholestasis. In fact, you use, or you induce, I should rather say, two types of biological, pathological responses, which are shown here in the yellow box, and that really would be the adverse outcome. But at the same time, you also induce here what is it, pink or the purple box, if you will, and that would be the homeostatic adaptive response. And again, that is something that is pretty special for cholestasis. It's not that manifested in other types of toxicity, but nevertheless, it also dictates the final outcome of the AOP. So that's why it's so important to have this adaptive response included in the AOP. So these adverse events or this adverse response, well, this is characterized by the induction of, I would say, the usual suspects like inflammation, oxidative stress, cell death. All that then results at the organ level in the presence and the release of a number of enzymes in the serum, which is typically how cholestasis is being observed and based on which that the diagnosis is made in clinical settings like AOP, for example, alkaline phosphatase. But at the same time, you also have this adaptive response that is induced and that relies on the activation of these nuclear receptors, which then triggers a whole bunch of transcriptional changes that all are targeted towards, basically counteracting this yellow response, if you will. So getting rid of bile acids, that's what we want to have done here with this adaptive response. This not only leads to the increased presence of bilirubin in the urine, but also in the serum, and this is what we call jaundice. So this typical yellowish pigmentation of the skin and also of the eyes. And finally, please have a look again at these key event relationships. There is a color code here. I'll not go into the details. If that were to be of interest to you, I would be happy to share the publication with you. But in essence, these key events, they relate to inductive inhibiting effects and also direct and indirect. All right, so we talked about AOP development. We also talked about the AOP principles that should be applied during the development and the application. We also talked about AOP evaluation, so assessment. But of course, the real deal is to figure out in practice if an AOP, which after all is a hypothetical construct, if that makes sense from the practical point of view. And this is what we like to call AOP validation. And I immediately want to stress here that this absolutely has not a single regulatory meaning. So we are not talking about validation of methods. This is a purely scientific or a research exercise. There are no strict rules on how to scientifically validate an AOP, but a very common procedure would be to test the robustness. And I would like to exemplify this by using the example of the cholestasis AOP. So this is work that my group, in collaboration with other group, has been doing over the past few years. Again, this needs to be adapted on a case-by-case basis. In the case of the cholestasis AOP, the first thing you want to do if you want to assess robustness here was to see to what extent it actually is applicable to different types of cholestasis. So remember from the introduction, there was intrahepatic cholestasis, and that is typically the kind of cholestasis that is induced by chemicals, but there is also extrahepatic cholestasis, so where the bile acids accumulate outside of the liver, so in the gallbladder, for example, which can be induced by, tumors, for example, these kind of things. So we wanted to check out here if the AOP that we came up with can be applied to both types of cholestasis. The way we have been doing this was modeling in vitro and partly in vivo. So the in vitro setting was a very simple HPRG cell culture. I do not know to what extent you're familiar with that, but well, it's now considered as one of the, let's say, benchmark in vitro models for testing cholestasis. We selected a number of compounds of which we know that they induce as side effects cholestasis. Yeah, I will not go into the other conditions here. But that was the model for testing intrahepatic cholestasis. We also wanted to test extrahepatic cholestasis, and that was based on an in vivo experiment. I want to stress here that we did not do in vivo experiments ourselves, but actually we were in a very fortunate situation of reusing samples that have been generated from a collaborating lab that actually set up this in vivo model of extrahepatic cholestasis for another type of study, but they still had the samples left, so we could reuse them. And the model that was used there was the so-called bile duct ligation, which is a known model for extrahepatic cholestasis. So to check whether the AOP actually made sense, we did quite a few of analysis. The most important one was a classical transcriptomics analysis. If you ever will or want to validate an AOP yourself, the first thing that you want to do, especially in vitro, is to show that the MIE, the starting point of the AOP, is there and that it's functional. That's really important. So not only showing that it is expressed, but also that it is functional. I remember that one of the most important MIEs in the AOP on cholestasis would be this bile salt export pump. So we showed that it was expressed, but we also had to show that it was functional. And again, I will not go into the details here, but this was done using this probe called CLF. So have a look here at this second column. So the first two rows are the control condition. You have this punctuate pattern, and this is what you want to see. Actually, this is this fluorescent CLF that has been pumped out from the cells, showing that BSAP is functional. But this is very different here in the lower three rows, which is where the cells have been treated with cholestatic compounds, so you don't have this function pattern anymore, which shows that BSAP is dysfunctional, which indirectly shows to you that BSAP actually is there in the cell culture system and that it's up and running. I will not go into the details, but here you can see the summary of the transcriptomics analysis. So we wanted to compare the setting for intrahepatic, so chemical-induced cholestasis versus the extrahepatic kind of setting, if you will. So remember that in the AOP, we had two responses, the adverse response and the adaptive response. We had a look to most of the key events. The message here is that we were able to reproduce most of the key events for intrahepatic cholestasis, but not for extrahepatic cholestasis, which tells you that the AOP cannot be used for this type of extrahepatic cholestasis, but only for chemical-induced cholestasis, which is essential to know, of course. Making use of omics analysis, you not only are able to reproduce what you already know, so reproduce what was predicted by the AOP, but also to identify potential new key events. And remember that we talked about the AOP principles, and the very last one was that an AOP is a living document. So it should be updated by, feeding in new data. And this is exactly what we have done. At the time of analysis, but that's already a couple of years ago, there was nothing known about types of cell death involved in this type of cholestasis. By means of this analysis, we showed that specific types of cell death like autophagy and necroptosis, were involved. And this also allowed us to update the AOP. So remember the AOP I showed to you with the yellow response, if you will, and the purple response. We decided to keep it simpler to make it fit for purpose, and I will be coming back to this further on. So this is again a more simplified version of the AOP, but still with the most important key events captured. And also here, as you can see in orange, including also the new key events that we have identified. So that was a first example of how you can test the robustness of an AOP to validate. The second aspect here of AOP robustness testing related to the type of chemical. So remember that we have tested all of this and that we have optimized all of this with pharmaceutical drugs, which is pretty much the low-hanging fruit because we typically have a wealth of data of pharmaceutical drugs, which is usually not the case for chemicals that come from other applicability domains. So what we did here is we did a screening of literature, but also of safety dossiers. Every piece of data that was actually relevant here. So we looked for chemicals from other applicability domains for which we have some clues that they might induce cholestasis. But of course, we did not have the full-blown kind of set of data that we would have for pharmaceutical drugs. So we ended up with a number of chemicals, as you can see here, that come from the food sector, but also from the cosmetic sector, of which we know from animal studies that they might induce cholestasis. We went through the entire same procedure as I exemplified in the first step. So again, we exposed these cells in vitro to these compounds in a number of conditions. We did transcriptomics analysis. We checked for the most important key events and a bit of an excerpt of the results here. You can see here. Bottom line is that this tartrazine, which is a dye that is actually being approved, it is approved as a dye for example, used in foodstuff, that it would, at least based on the AOP, would be predicted as an inductor of cholestasis.
Mathieu,
yeah,
we have a question on the chat. The question is, how to distinguish adaptive and toxic responses?
Yeah, you can do this basically by setting up, experiments in which you try to identify the rate limiting step. So it's all about the sequence of the events. What you can also do is to, let's say, just inhibit a number of the events, and then still see if the AOP would take place, and then you could also distinguish between an adverse or an adaptive response. What we have seen for the AOP is that in practice it was not that simple. It was not that, as we hoped it would be, in the sense that time-wise, you have at the very same time the induction of the adverse event and also of the homeostatic adaptive response. But I know for several other toxicological effects outside of the liver, that would be different. So you would have the onset of the adaptive response a bit later, and that would allow you also to distinguish between both events.
Does this relate also to, let's say, concentration effect relationships that you can distinguish between the two things?
No, not based on concentration. It's more time-wise. You need to look into it. Again, the best way to do this is just to, basically try to get rid of a key event. You can do this, for example, by means of siRNA. If you want to quench the expression of a number of mediators of that event, or a simple functional inhibitor of that particular event, and then to see whether the AOP as such still takes place, to what extent it actually does. But you cannot pick this up by playing around with the concentration of a chemical. No.
Because that is, in many cases, the essence of, let's say, a toxicological risk assessment. Huh? It's playing around with the concentrations. Maybe you'll come back to that later.
Yeah, that's actually what I will do at the very end. So here, bottom line is that the tartrazine, which is approved now, it is on the list of EU-approved, food dyes that, at least based on the AOP, that it would induce this cholestasis. And then a final example, just to complete the study, was testing robustness at the level of the in vitro system. And that's a whole webinar on its own, liver-based in vitro systems. I mean, the gold standard still is primary human hepatocytes, especially if you culture them in a spheroid configuration, so 3D. So we tested this. We wanted to check if that makes a difference. So compared to a simple HIPRG cell culture system. Another thing that we wanted to check here, and I think this comes back to what was just asked, is whether it's really necessary to culture for long term. That would be 28 days here, especially to see whether you would see a better and a more manifestation of this this homeostatic adaptive response. And that was at the time when we set up this study, was something that was being discussed in literature. There was a group at the Karolinska Institute that claimed that you could only pick up some special cases in terms of cholestasis induction by having this very long-term testing regime, so up to 1 month. So we wanted to study this. So in terms of exposure regimes, we have included 7 and 28 days. Again, that is being published. It has been published. It's open access. Just send me a mail if you don't have access to it. Anyhow, I can send it to you. I will not go into the details here, but just the take-home message was that indeed, for some of the compounds, we could pick this up. But we did not need 28 days. So again, that might be an answer to the question here. You need 7 days, so that's something that you would need, but you don't need like 1 full month in order to have this adaptive response being manifested, whether or as you can see here as well, we used again the AOP for benchmarking purposes. What we also noticed here is that it does not really matter if you use a more complex primary hepatocyte 3D culture or a simple 2D HIPRG monolayer culture. I don't want to generalize this because for other toxicities, it might be very different, but at least for cholestasis, that was not necessary. And it also brings in a second advantage because several of the assays, especially functionality assays, if you want to test cholestasis at the activity level, are simply not compatible with a 3D configuration. So that would be an additional advantage here, which again allowed us to update the AOP, again, bringing us to the 5th principle of AOP, so saying that it is a dynamic document. And actually, we published this last year. We took advantage of artificial intelligence. So the very first version of the AOP, and I think most of the AOPs out there, are the results of manual collection of data, which bears the risk, of course, that you would miss out and overlook some data because there are so many out there. So that's why in the context of a European project I currently coordinate, namely OnTox, we used this SysRef, which is basically a machine learning tool to update the AOP. Again, don't have time to go through it, but the publication is there. It's open access. We started off with more than 6,500 people. We recruited 10 experts, cholestasis experts, and that includes myself. And we asked them to manually go through 10% of the paper, so about 600 papers, to extract relevant data that could be fed into the AOP. And at the same time, some very clever people actually were teaching the machine, so machine learning, on how to do this. And we then left over the remaining 90% to the computer to retrieve all of that information. And we also took advantage of this to kind of visualize the result of the Taylor-Bradford Hill criteria assessment. Again, this is a figure that may come across as very trivial, but here again, I can assure you that, and I'm not exaggerating, there is 1 year of work hidden behind all of this, not of course full-time work, but it shows you the result of this. The good thing is that it kind of kept the original structure of the AOP, in the sense that the importance of the different key events was confirmed. But now this is fully confirmed by at least all of the relevant information that is out there. Again, please go through to the paper, but just to mention here that, for example, the thickness of the edges gives you a reflection of the evidence that is out there in terms of supporting the key event or the key event relationship. It was made fit for purpose, and the purpose, as you will see further in the presentation, would be to serve as the basis for setting up an in vitro test battery. And this brings me actually to the final part of the webinar, AOP applications. So very often, if I give this kind of webinar, then people tell me, well, Mathieu, it's very nice what you're telling there, but it seems to be a very academic kind of exercise. So what can you do with an AOP? Which is a relevant question, I would say. Well, I think there are some very nice examples in literature, also at different regulatory agencies, of how AOPs can serve the purpose of toxicology, but in particular risk assessment. Again, I will not go into the details here. That really is a course on its own, but you can use them for chemical grouping, chemical prioritization. They can be considered as the backbone for setting up these IATAs. This figure, what you're looking at, is very important because it illustrates again one of the very important principles of AOP, saying that an AOP should be fit for purpose. So already when you start to develop or optimize an AOP, you should have a very, very good and very well delineated idea of what the purpose of an AOP will be, because this will dictate the maturity, which then in turn will also determine, let's say, the application. That's important. So for example, a putative, so a very simple AOP can already be sufficient for chemical prioritization, while you would need a full-blown AOP network, and preferably quantified if you want to use it for IATAs, for example. Now, I would say the most common application or use of an AOP would be to use it as the basis for setting up in vitro test batteries. And this is exactly also what my team is doing in the context of a couple of projects ongoing. And this is the workflow that is typically followed for any kind of AOP, so not only for liver AOPs. So you would start off by, either developing a new AOP if an AOP is not already out there, or optimizing an existing one. So by relying on all kinds of data that would be relevant, and preferably you should assess it according to the Taylor-Bradford-Hill criteria. And then you have a kind of a mechanistic compass or a roadmap, if you will, for setting up an in vitro test battery. So where you have different assays, each of them that are mechanistically anchored in an AOP. I would like to exemplify this shortly for the case of cholestasis. Actually, we embed this in a tiered testing approach. So let's say that you would be working in a company, and that one day your boss enters your office and he or she would say, like, well, this is this new molecule. We are interested to bring it on the market, but we need a safety and a toxicological assessment. Well, this is the molecule. Here you have the structure. You have some physical chemical data. We do have very, very limited animal data. And I would like to ask you if you would be able to assess its probability to induce, for example, liver cholestasis. Good luck to you if you're not allowed to use animals for this, and well, try to pull this off. So how to start with this? Well, the first thing you always can do is just having a look at the chemical structure of the chemical along with the physical chemical profile. And this can already give you some clues about, the potential to induce certain types of toxicity. Think about things like QSAR, these kind of things. Certain physical chemical properties have been associated with a cholestatic potential. You can also have these very nice, in silico modulations, like simulation in this case of drug transporter inhibition, molecular docking, these kind of things. Very nice. That's a first, let's say, kind of a piece of information. The second two tiers would be fully relying on in vitro testing. So there you could set up, could be a very simple, system to start off with, like this HIP-RG cell culture. So you can expose the cells to the molecule of interest. And then if you have access to this, you can do a holistic approach, which typically is a microarray RNA sequencing. You can also do proteomics, of course. And the idea would be is to really, pick up the entire transcriptomic signature that is also depicted in the AOP, which you can see in the dashed box on the right-hand side. So that's nice for several types of toxicity, including steatotic liver toxicity, but also extra hepatic toxicities. A transcriptomic approach could already give you a predictive power of, let's say, 90%, which is really nice. But for very complex types of toxicities like cholestasis, only doing transcriptomics gives you a predictive value of something like 60%. And the reason for this is probably, if you think back about, the AOP, is because of this very pronounced homeostatic adaptive response. So that's why this complicates, basically the prediction of this. You can get this improved by having this second tier followed by a third tier, which is what we call an atomistic approach. It's again a very fancy word, but in practice, it comes down to setting up, like a series of assays, each of them that are mechanistically anchored in at least one key event. So this is what you're looking at here. This is just exemplified here. I don't claim here that this would be the in vitro test battery for cholestasis. It's just to give you an idea of how this works. Actually, as we speak, we are playing around with the composition of these assays in order to have the highest predictive value. Just at the end, coming back, or shortly, some words about the second tier, because we do know that several companies that have limited resources do not have access, for example, to things like RNA sequencing or micro arrays. So several companies do typically still do PCR kind of analysis, and this is something that you can do. So you do not necessarily have to pick up the entire transcriptomic signature. What you could do is let's say, identify a number of indicator genes. And there are two ways to do this. So you could identify these genes by means of artificial intelligence, machine learning, and so on. Actually, we very recently have done this. Some very clever postdoctoral researcher in my team, by going through a lot of databases, applying artificial intelligence, came up with this list of 13 genes that are indicative of cholestasis, but that cannot be used as a standalone indicator. So you still have to use it in combination with like measurements at the functional level, if you will. The other option, which is not mentioned here, but that is that you also can manually select genes, and that would be if you have expert knowledge on this, and if the AOP is not really that complex. Actually, we recently done this for the case of liver steatosis, so you don't necessarily need to apply artificial intelligence. So with this, I'm at the very end of the presentation. So if you were to have any questions now, but also later on, I'm always happy to to discuss. So don't hesitate to reach out to me and ask questions. And of course, these slides can and will be shared with you. Thank you very much for your attention.
Okay, thank you, Mathieu. At least there is one question on the chat. It is about when you analyze AOPs through transcriptomics, which filtration do you use for selecting different expressed genes, such as p-value, etc.?
Yeah.
This criteria depends on the researcher, of course.
Yeah. So we played with several things. So we, let's say statistical kind of parameters, but also biological parameters. So have a look at our publications, because this is exactly what is explained in this. It's not one single kind of procedure for all. It really needs to be adapted case by case. So depending also on the outcome and what would be the application of the outcome. But there are a number of let's say, gold standard kind of practices indeed. We don't have to go into the details here right now, but usually we go for the statistics-based strategy.
Mathieu, there was a question also in the earlier webinar on Tuesday
Yeah.
When we are talking about mechanisms of action or MIEs, etc., how do we deal with epigenetics?
Yeah. Well, epigenetics is indeed, you know, let's say more at the post or pre-transcriptional level. This is something that can be included in an AOP as what we call a modifying factor. So epigenetics would typically lead also to adaptation, but also to the more personalized way of how people respond to toxicants. It's difficult to capture this in an AOP as a key event because, again, this is really personalized, but you can have it included as what we call a modifying factor. And this is also in compliance with OECD guidance. But again, if you were to fully include this as a key event, it would not be an AOP anymore. It would be a mode of action. Yeah.
Yes. Yeah. Yeah. Okay. Well, I don't see any other questions in the chat. So thank you very much, Mathieu, for this presentation. All right. If we're all back, it's now time for the presentation by Nynke Kramer. Nynke, I see that your presentation is already on the screen. Nynke is a associate professor of toxicology at Wageningen University. In the past, we worked together at Utrecht University. She is an expert in the field of kinetic behavior of compounds. And she will talk about PBK modeling and also about how to use this in a so-called quantitative in vitro in vivo extrapolation. Nynke, the floor is yours.
Thank you very much. I hope you can hear me okay.
Yes.
Great. So thank you all for making it to this webinar series. And just a heads up here. So I'm going to really just give you an introduction to, chemical kinetics, or the kinetics of a compound in the body, and how we use that knowledge to make PBK models, and how we use PBK models for quantitative in vitro to in vivo extrapolation. So really it is an introductory talk, but I'm very happy to talk about it much longer than I have these next 45 minutes, so do reach out if you have questions. This is just a very quick declarations of interests and conflicts of interest. There are none that I can see, but just a note that I'm employed by Wageningen University, and I do have a couple of grants from the EU, from the Dutch Research Council, and also from IFSA. Bas already explained this to you. Just a quick recap of who I am. So I'm an associate professor in toxicology at Wageningen University, but I have spent much of my time at Utrecht University, both for my PhD, for my postdoc, and for my assistant professorship. I am actually an environmental chemist by background, so I did environmental chemistry, but the processes that happen in the environment are very much similar to the processes that happen in the body, at least when it comes to the movement of chemicals. And that's why it's a very neat combination to have both that environmental chemistry and human physiology as a background. So in this webinar, I'm going to explain how these PBK models are used in quantitative in vitro to in vivo extrapolation, or QIVIV, and how that then fits in the framework of next generation risk assessment. Now, I do realize that for some of you, this might be gibberish, but fortunately, Bas and Cyril and Mathieu have already covered quite a lot of it, and I will just put it all, wrap it all together and include toxicokinetics here. So the outline of my presentation is as follows. I'll be answering basically these 4 questions. This includes, what's wrong with the way that we perform toxicological risk assessment now, and I will focus on toxicokinetics, of course. Then I'll try to answer the question why it's so important to look at in vitro toxicity assays. This was covered by Bas yesterday. But what I'm going to answer predominantly is the question, why are in vitro assays actually useless without the knowledge of toxicokinetics? And so once you have that knowledge of toxicokinetics, the question that can then be answered is, why do we need PBK models in this context of next generation risk assessment or toxicity testing in the 21st century? So let's start from the very beginning. Traditional chemical safety assessment, as you're probably well aware, we normally test this on animals, on rodents. Most of the cases, we assume that, this is the rodent here, and we assume, depending on the case, but we assume that humans are 10 times more sensitive than the rodent to toxic effect, to toxic perturbation, and that the individual and the sensitive individuals like infants or the elderly are even more sensitive than the average human, and so we divide by another factor of 10. So this is how we crudely see risk assessment. At least we have done so in the past, and we use these uncertainty factors because we know that there's a difference in the way that an animal deals with a chemical, and that can be in two ways. The way that the body reacts to the chemical, so that would be toxicodynamics, or the way that the chemical, the body moves the chemical through it, and so that's toxicokinetics. So I've put a next slide in there too. So if you take mercury, for example, when it gets into the brain, but also the kidney, it will disrupt neurotransmission there, and that's why it's neurotoxic. This we would call the toxicodynamics of the chemical. However, the chemical has to actually reach the brain, and so for the chemical to reach the brain, the body has certain processes that it undergoes, and this is what we call toxicokinetics. And basically, toxicokinetics is a function of 4 major processes: absorption, distribution, metabolism, and excretion. When we talk about foods and chemicals in foods, normally they're taken up as food. So the chemical comes in through the gastrointestinal tract, then goes through into the liver, where it can be metabolized. But of course, other chemicals you can inhale or you can take up dermally. Once it actually gets through the intestine, through the liver, it can get into the general circulation, and there it's distributed to the different tissues. It can be metabolized usually in the liver, but also in the gut and certain other organs. And once that's the case, once it's in the circulation, it can also be excreted, again, as biliary excretion or through the urine, for example. So those are the 4 processes that determine toxicokinetics. And they do differ a lot between animals and humans, but also between you and me. And this is why we use these uncertainty factors. But what's wrong with the way that we do toxicological risk assessment when we base it so heavily on these animal tests is that these animal tests are black box models. There's a lot of uncertainty when it comes to species extrapolation. There's a lot of variability, and we usually take mixtures, we don't take mixtures into account. And that brings along with it a lot of the ethics that are associated with animal testing. And if you look at the Cosmetics Act, we're actually not allowed to use animal testing anymore. And an important aspect to do away with these animal tests, or as much as we can, is really to their time and money resource rich experiments. And so this is also why, as Bas said yesterday, in vitro assays are so incredibly powerful. We can use human tissue for this, and so we somewhat circumvent this species extrapolation issue. It's ethical. It can be done in high throughput, but what I find most important is that it gives us a mechanistic basis of understanding the toxicity of compounds. And this is also what's been put out by the US in its the National Research Council. There they have the toxicity testing in the 21st century report, this landmark report in 2007, and it's been updated in 2017. But there's been a relatively slow uptake of these in vitro alternatives. One interesting case that can exhibit why this uptake may be a bit slow is to look at the data that has been produced in the ToxCast program. So this is short for the Toxicity Forecaster. It's part of the toxicity testing in the 21st century program, or TOX21. And it's a collaboration between the US EPA, the FDA, and the National Institute of Health. And what they've done, they've screened 10,000 environmental chemicals, for which there's not always a good amount of information when it comes to toxicity, and they've used these high throughput robotic in vitro assays. So it's a very, very extensive program, and they've screened for any of those potentials to disturb these adverse outcome pathways that were discussed by Mathieu. But this is what came out, at least this is what used to come out back in 2010 when they started. So on the x-axis here, you've got a whole list of chemicals that they tested in this some 700 plus in vitro assays. And on the y-axis here, you have the median effect concentrations in each of these in vitro assays. And I don't know about you, but it's very hard to discern which of these compounds are a greater risk for you and me than and which ones are not. So they all seem to have relatively similar effect concentrations in these in vitro assays, at least the ranges. And this, well, we will argue here, this is because we're missing a very important part of what determines toxicity, and that is toxicokinetics, not just what the chemical does to the body or to a particular receptor, but what the body does to the chemical. Of course, and fortunately, Mathieu Vinken has already explained this quite in detail. One part of the extrapolation issue that we have is that we need to translate these molecular perturbations in these in vitro assays to, a clinically observable effect. So, for example, a change in one of the receptors that's associated with cholestasis, like BSEP. How much inhibition do you need in that of that transporter before you see cholestasis on a whole organism level? And this is where adverse outcome pathways are incredibly valuable sources of information, or it's a very valuable framework. So in theory, it translates that y-axis on a dose response curve. So it translates the amount of change in a cell system on a molecular level to a whole body. And so, for example, the changes in cells to the number of people that show liver disease. But of course, we're still missing, and we're not accounting for toxicokinetics. So we still have a dose response curve where on the x-axis, it's a concentration in these in vitro assays. So the question really is, how do we translate that to, for example, an oral dose, the amount that you and I would need to take in to get that level of change in the liver where the toxicity occurs. So what we need to understand is how we go from an external dose to an internal dose, like a plasma concentration, to a target dose. And if you really pull that all the way through, you would want to have a concentration that's inside the cell where the effect is occurring. So that's the biologically effective dose. And to do that, that process of that translating that in vitro concentration to a bioequivalent human dose, we call that quantitative in vitro to in vivo extrapolation. So the formal definition here is the estimation of chemical exposures producing target tissue exposures in the species of interest. For us, that's often the human. And that dose should be equivalent to those associated with the effects in these in vitro tests. So how do we do this? So how do we make that translation? Well, first, it's really important to understand the physiological processes that drive toxicokinetics. Once we know that, we can develop and evaluate physiologically based kinetic models. These are these PBPK models. And once we have a good PBPK model that we trust, we can then edit that PBPK model and translate it into what we call a reverse dosimetry model. And we can use that for quantitative in vitro to in vivo extrapolation. Again, here, to some of you, this is gibberish, but I'll go through each of these steps. So let's look at the toxicological the biological processes that drive the toxicokinetics. So that's the absorption, distribution, metabolism, and excretion of a compound. Normally, at least when we talk about chemicals in food, it's ingested, it comes into the gastrointestinal tract through the portal blood, goes into your general circulation. It can also, of course, be inhaled or dermally taken up. Once it's actually in this general circulation, from there it gets distributed all throughout the body. It can then pass in through the extracellular fluids and then into every tissue of our body, at least that's connected to the circulation. Or it can go into non-target tissues. This would be tissues like fat or every other tissue that there's no toxicity in, or at least the toxicity that you're looking at. Of course, not every part of the chemical that is in your gastrointestinal tract will actually be absorbed. Much of it can actually just go straight into the feces. But if it does get absorbed, it can actually go into from the portal blood straight into the liver and be metabolized, go through the bile and then be right back excreted into the gastrointestinal tract, which we would call biliary excretion. If it does get from the liver, it goes, or from the portal blood into the liver and go into the general circulation, there it can also be excreted. And usually that can go through urine by passing the kidney into the bladder. So those are major excretion routes, the biliary excretion route and the urinary excretion route. But of course, there are also some minor excretion routes for some compounds that actually are quite significant. For example, if you're looking at pregnant ladies, or sorry, at breastfeeding ladies, the secretory structures like breast milk might also be a significant route of excretion of a compound, at least a very relevant one, because it means it decides on how much gets into the milk and how much the infant is then exposed to. And of course, everything that you can inhale can also be exhaled, so that's another route of excretion for you. So that is toxicokinetics, at least the biology behind it, in a nutshell. So normally, and you might ask the question, it's like, why don't we just then use the classical compartmental models that we use in pharmacology a lot? Well, the reason for that is because they're empirical bases, and the issue is they require animal testing. So just as a quick recap, because they do help us understand PBK models, when we talk about compartmental modeling, usually what we do, we expose an animal like a rat intravenously first and then later orally to your compound of interest, and we tap a bit of blood or look at the urine, and we absorb, we calculate the amount of the chemical in the blood at a different time points. So you've got your time plasma profiles, and from there we can fit a one-phase decay function, and we can calculate the rate of elimination, or the rate constant of elimination, and from there we can calculate the fraction absorbed, where it gets distributed and eliminated, and then set the dosing regimen that was necessary for a particular drug. But it's not very helpful when it comes to a next generation risk assessment, and this is because it's not physiological, because we're using the animal and it's an empirical model, right? And it relies obviously on these animal tests. And what's really important to note when it comes to these models is the rate constant, so the elimination rate constant is assumed that constant with every dose, but we know that's not true because it doesn't take into account the dose limiting processes. It only focuses on one exposure route. For example, the oral exposure route, you'd have to do the test again and refit it if you want to look at another exposure route. And of course, every individual is different, every species is different, and it doesn't take these differences into account. It assumes that, the rate of elimination is similar in your test animal as it would be in you and me. And importantly, when it comes to next generation risk assessment, where we want to really focus and have a mechanistic understanding of a compound, and we focus on the concentrations in tissues, which we then translate, which we then mimic in these in vitro assays, is that we cannot predict the time profile, the concentration time profiles in specific tissues. We assume it's going to be quite similar across tissues. So this is why we use physiologically based kinetic models. These consist of a series of mass balance differential equations which simulate the transport of chemicals between tissues. So bear with me, but basically what it is, it's a whole, it's a suite of these compartments, and they actually represent real tissues. So, you'd have a constant, if it, you have your intestinal compartment, you'd have your liver compartment, and all these compartments are linked together by the venous blood and the arterial blood. And the chemical would come in here, and you estimate the change in the concentrations in each of these tissues, and you do that by using a suite of these differential equations that look daunting at first sight, but I will take you through each of them, and then hopefully that makes sense a little more than it might have in the beginning. But basically, the amount of a chemical in a particular tissue, like the brain, is a function of how fast the blood flows through that tissue, and what the concentration is in that blood, both in the arterial blood and the venous blood that comes out of this tissue. And the amount of the brain is obviously, the change in the amount of the brain is obviously a function. The amount in the brain at any time point is obviously a function of the integral of the change in the amount in the brain. And the concentration in the brain is a function of the amount in the brain and the volume of the brain. And then how much goes from blood into a tissue, that's dependent on the partition coefficient of a particular chemical. So that's how much it loves sitting in tissue compared to a reference phase, in this case the blood. So the higher the partition coefficient, or the tissue blood partition coefficient, the more the chemical likes sitting in the tissue compared to sitting in the blood. So it's a constant. And so that, in a nutshell, is the set of equations that it actually goes and feeds into each of these compartments. And if you look, having explained this very briefly, there are really three types of input parameters. So you have your exposure parameters, for how much is in the gut, so how much is in the food. You have your organism properties, so that's the blood flow rates to the different tissues, for example, but also the volume of each of the tissue. And most importantly, you have these chemical properties. And these chemical properties can be something like the blood partition coefficient and the tissue blood partition coefficient. But I've listed the important ones that come into these PBPK models here. So for absorption, you can use, again, these partition coefficients, for example, in the lung, but you can also use permeabilities. So that's the permeability constant of a chemical across a certain barrier. You can use distribution parameters like these tissue blood partition coefficients I talked about, but also the unbound fraction in plasma, and again also permeabilities if that's the limiting step. Then of course there are metabolism parameters in these models. So think about the clearance, the intrinsic clearance, for example, of a particular chemical, or better yet, the maximum velocity, the Vmax, and the Michaelis-Menten constant, the Km of a particular chemical for metabolism in the liver, for example. And then, of course, excretion is usually a rate like glomerular filtration rate, but it can also be, if it's excretion through metabolism, they can or transporters, you can also use these same Michaelis-Menten kinetics parameters. So when you have the model like that, and it's established with the differential equations, the question is simply solving it. And this is a really neat aspect of PBPK models. It really simulates. So it's not an extrapolate. It's not like compartmental models where it's empirical. It's really a simulation of the concentrations of the chemicals in each of these tissues that you've modeled here. So this would be your concentration in blood, and these would be the concentrations in different tissues, for example. And the neat thing is, because it's physiologically based, you can, in theory, make any type of animal or individual that you may like and that you have data for. So when you make these PBPK models, they really consist of what I say 6 steps. So you first have to decide on a model structure. So what tissues are you going to include, for example? How are they related to one another? The second step is that you need to translate each tissue into a set of equations, mathematical equations that I briefly showed before, but I'll go into a little more detail. And then, of course, you have to define these parameters, the volumes of the tissues, but also these chemical specific parameters. And once you've done that, you can solve them. And that means solving means creating these concentration time profiles in each of these tissues. Of course, it would be good to validate it. Am I predicting or am I simulating real simulations? Am I simulating real life? So am I predicting what I should be predicting? Is the concentration in the plasma indeed the concentration in plasma that we see in a particular animal or in human patients? And then once it's validated or evaluated, we can apply the model. And this application specifically would be for quantitative in vitro to in vivo extrapolation. So let's look at the first step here. So when we look at a model structure, what is really important here is that it's a complete organism. It's a complete human, where each of the tissues are obviously linked to one another. And it should also be as simple as possible and as complex as necessary. So don't include too many tissues, because you would also need all the parameters for that, and it may not actually be very important. But what you would need, at least, when you decide on the type of tissues that you want to include, it should have the absorbing tissues that are relevant, the connecting tissues, so that would be your blood in most cases, but sometimes also lymph. You would need your target tissue. So if you're interested in neurotoxicity, you would include the brain. It would need excreting tissues, so that's usually the liver and the kidney. You would also need to include storage tissues. Fat is a very common compartment to include in these PBK models, because a lot of chemicals, particularly the lipophilic ones, will store in these tissues. And then, of course, it'd be good to include the tissues that you actually have validation data for. So it could be biomonitoring data, or if it's been tested in a certain setting in animals, that would be good to include that particular tissue. And everything else is lumped together in a rest of body tissue. So every other tissue you're not interested in, you would lump into this massive rest of body tissue. Usually we do distinguish between two rest of body tissues compartments: the slowly perfused tissues. So think of muscle here. The level of the accumulation of a compound in these tissues is quite slow, just because the blood flow rate to these tissues are slow. And the richly perfused tissues. So these are tissues like your heart and many of the liver, etc. They're very richly perfused. And so that's your model structure for you. The next step then is to translate it into mathematical equations. For your reference, but you can see the slides later, I've listed all the equations that really just come into these models that will go into each of these tissues. But I'm not going to name them all and I don't expect you to understand them if you've never seen this before, if it's the first time you see it. But basically, the amount of the tissue is a function, or the concentration of a tissue is a function of the amount of tissue divided by the volume. You include this partition coefficient, the tissue blood partition coefficient, when we translate a concentration in blood into a concentration in a particular tissue. And we use Michaelis-Menten kinetics, ideally, to estimate how much of a compound gets metabolized over time. So once we've defined every compartment of a PBK model with these differential equations, it's really important to find the right input parameters. But how do you do this when you don't actually have an animal to test, to get these parameters? And this is where I'll spend a little more detail in a bit. I've explained this briefly before, but just as to summarize, there are really three types of parameters that you need to get and to input in these PBK models. And they include these exposure properties, right? The formulation, the concentration or amount of the chemical that's in the gut, but also the organism properties like the blood flow rates and the volumes, and also the chemical properties that are obviously associated also with the organism properties. And these are the ADME properties: absorption, distribution, metabolism, and excretion properties. So the unbound fraction in blood, the partition coefficients to different tissues, and these permeability constants, for example. So where do we get these parameters, particularly these chemical specific parameters? And these you can use in next generation risk assessment. At least you can use two types of tools. The first one are in vitro assays. So unlike the toxicity assays, the in vitro toxicity assays that I discussed before, these are the ADME assays, the absorption, distribution, metabolism in vitro assays. So I've got an example here. This is the KECO2, schematic of the KECO2 permeability assay, where you add the compound in the apical compartment and you observe the speed at which it passes the membrane, the intestinal membrane here, into the basolateral compartment. So, and from there you can estimate an apparent permeability, which you can then translate into a flux and use that in your PBK model. Another option, of course, is to use QSARs, quantitative structure activity relationships. They usually relate a chemical structure or chemical property, like the lipophilicity of, like the octanol water partition coefficient, for example, to another property. So it could be toxicity property, but in our case, we're looking at properties like the unbound fraction in plasma or the tissue partition coefficient of the particular chemical. And this is a very classical example of one of these QSARs that is often used in PBK models, from Joost de Jong in 1997, where they found a relationship between the optimal water partition coefficient of industrial chemicals and the fat blood partition coefficient. And you can use this regression to then calculate the fat blood partition coefficient. Again, I'm not going to read this out, but this is for those of you who are interested in actually working with these models yourself. I've listed the major QSARs that can be used as input into these PBK models. So you've got, and I've separated them into categories: the absorption parameters, so apparent permeability, for example, across the gut; the distribution parameters like the unbound fraction in plasma; the tissue blood partition coefficients and the different QSARs that can be used to calculate the tissue blood partition coefficients, and most importantly here too, the clearance, the metabolism parameters that go to PBK models. Now, I also will say that there are no good intrinsic clearance, or at least quantitative intrinsic clearance estimating tools or QSARs available. Although I would say that with the advent of artificial intelligence and making better databases, I think we are coming close to developing some good ones. But for now, I would say please use in vitro assays to estimate your intrinsic clearance of a compound. So if you look, the way you can do that is usually using hepatocytes or subcellular fractions of the hepatocyte, or if you're interested in extra hepatic metabolism, then you would also use tissue like the intestinal tissue, and you can add the compound and see how fast it gets metabolized in time. You would take an extract, use chemical analytics to derive concentration response curves, concentration time profiles of your compound. And this is just the equations how you would calculate the intrinsic clearance and move that from an in vitro intrinsic clearance to an in vivo intrinsic clearance. And this is then used in PBK models. There are also some, and this is what we're particularly working on also in Wageningen University, are apparent permeabilities to understand the flux of a compound across a barrier like the gut, the enterocyte barrier. I've listed the major models that you can use. The most commonly used model to estimate apparent permeability that then feeds into PBK models is the KECO2 permeability assay, as I explained before, but there are several others, and we're also looking at the more complex models like the stem cell models or epiintestinal, for example. But I've already explained it's actually quite a simple process where you absorb the change in the amount of the concentration in the basolateral compartment, and from there you can calculate a permeability, an effective permeability in humans that include a fixed number of tissue compartments and have already integrated the QSARs so that they can automatically estimate chemical specific parameters like the unbound fraction in plasma or the tissue blood partition coefficients. And the user then just simply provides exposure and metabolism input parameters before running the model. And what I've done here is I've listed the major generic tools that are out there that you could use for this purpose. So the US EPA within the TOX21 program have developed HTTK, which is a very simple model. The UK Health and Safety Laboratory has a very neat model that's ARVIS that can be used for QIV. And then there's also relatively new, that's Plethum, which is run by Cytovation, and IndusChemFate, which might interest you, is actually a generic PBK model that runs in Excel and is quite good for a simple industrial.
Nynke?
Yes.
There is a question in the chat.
Mm-hmm.
Two, by the way. The one is related to this. It's the US EPA has a web-based IVE model.
Mm-hmm.
and to what extent this web-based tool is helpful for PBPK modeling or the other way around. And the second question is, do you have any experience using EFSA's TK plate?
Yes, so absolutely. So EFSA's TK plate, I haven't put on there because it just came out. But yes, we have recently in the last few months been testing TK plate, and it's I would say an impressive model, and particularly for those who within the food area. It's open source and it's relatively easy to use. So yes, TK plate is a great tool for, at least definitely for the simple first tier PBK modeling. HTTK runs in and the IVIV tool of the ICE tool of the US EPA is a beautiful source of a lot of information. So it's linked to Comtox, etc. The model itself, the PBK model itself, is actually it is really simple. So it's really as a first tier approach. It doesn't include a lot of compartments, etc. So that's just for you to be aware of. That's why I will say, so it's important to understand, you know, the model structure of each of these generic models to understand if it's relevant for you to use in your particular risk assessment setting. But again, TK plate is an impressive new addition to this list here. Did I answer both questions?
Okay.
Great, thanks. For those of you who are looking for for pharmacology and non-open source, the major generic models that are out there is SimSip Simulator, for that Sotara. GastroPlus is excellent for particularly oral absorption, and PKSim, which is semi-open source. So that's again something to be aware of. They're relatively complex PBK models. They're very, very well tested, but generally for drug purposes. Just to illustrate one of them, this is our own tool at Wageningen University, QIVIV tool. So if you type in qivivtools.wr.nl, you get this interface. You type in the physical chemical properties of each of your compounds here, of the compound of interest, and it asks you which QSAR you might want to use, but it includes an explanation. And then it runs it for you. So this is an online tool, and you can see these concentration time profiles in the tissue of selection that you've selected. But this is important to realize that, you know, like what I've just shown you are deterministic models. So it simulates a single person with every parameter defined as an exact value. But of course, you know, your body weights vary, the brain masses vary, but also, you know, like the speeds of the blood, etc., but also the chemical properties you might not be too certain of. And this is where we can use population PBK models. So instead of giving it an exact value, you can give it a range, and then use Monte Carlo simulations to simulate, you know, thousands of individuals, for example, to see the range at which you can expect the concentrations in each of the different tissues. So once you've used either a generic model or you've made your own model, you can start validating them. Now, validation is quite simple. It can get more complex, but I don't have enough time to go into detail here. But usually, you know, you run the model and see for a certain exposure scenario for which you have toxicokinetic data for, and then you see whether the chemical, the simulations overlap with the toxicokinetic data that you have, and then you refine the model until it works. That's validation in a nutshell. There's more to it, but I think this we'll keep it here, and I can explain more detail later. The reason for that I'm going quickly is because this is very important. The application of these PBK models for quantitative in vitro to in vivo extrapolation. I've listed a couple of like studies that where PBPK models are currently already being used in risk assessment. So it's particularly used to understand, dose-dependent effects. So when there's some sort of saturation process going on, so think about drug-drug interactions, for example, it's used to extrapolate from low dose to high dose and high dose to low dose. It's also used to study route-to-route extrapolation. How does the uptake of the compound, how does the kinetics of a compound change if you take it by inhalation as opposed to take it orally? So, and it also allows us to study additional exposure scenarios, for example. Then
There are two questions again on this chat. One is, how do I simulate drugs with unusual kinetics, such as Cmax appearing twice due to lymphatic circulation, etc.?
That's a very specific question, but in this case, you would include both your lymph circulation in your model as well as your blood circulation in the model, and so also when I sometimes we say it's very rare, but we sometimes see when there's enterohepatic circulation, you tend to sometimes have two peaks.
But how do you know that you have to take that in?
Yeah, there's no, that's a really good question. So for these unusual ones, so when it comes to enterohepatic circulation, let me keep a short answer. We're working on that.
Yeah. But well, one answer would be probably is if the validation is showing that you are completely out of the range, then you have to think about the possibilities, why that is.
Exactly. Yeah, exactly. So this is what but then it does rely on animal data or human data already.
Yeah.
So
about your physiological knowledge, so to say.
Absolutely.
Yeah, and the other question is, which are the input parameters predicted with the best performance by QSAR models based on your experience?
Yeah, so that's an excellent question, in fact, but that is very chemical class dependent, to be honest. Rogers and Rowlands and Bereskovsky, and so you have a couple of these QSARs that feed into, I can quickly go to your list here. And this, again, this is my my preference, right? So it's very much chemical dependent. So when you look, for example, at these partition coefficients, whether you use Joost de Jong's QSAR or Bereskovsky's QSAR or Rogers and Rowlands is really dependent on the class. What Bereskovsky and Rogers and Rowlands have as an added benefit is like they account for differences in lipid content and protein content in the different tissues to calculate these partition coefficients. So they tend to do quite well, at least better for ionized or ionizable compounds. But Joost de Jong, actually, that model has been trained on simple industrial chemicals, toluene and ethylene, ethanol, etc. And sometimes it seems to be better for the more slightly lipophilic neutral compounds. So again, it depends on the chemical and the chemical class that you're dealing with, which QSAR you should use. And the best way to find out whether is to check whether it fits the applicability domain. So whether, what chemicals have been used to train these QSARs and evaluate them. Does your compound of interest fit that training or evaluation set? If yes, you can trust the QSAR better than if it's not the case. So
Thanks.
Yeah. That's clear.
Go ahead.
Thanks. Okay, so I'm just going to move on here. So the issue is that we actually want to use it in quantitative in vitro to in vivo extrapolation, but we can also use it, and what it's been used for in the past already is for example, understanding species differences in kinetics and therefore toxicity of the compounds. You're understanding why certain individuals, like patients, are sensitive to a certain type of toxicity, for example, because their glomerular filtration rate is much lower. And that's why you see more need for toxicity for a drug, for example. And within the remit of EFSA and these risk assessment reports of foods, we can use them to establish chemical specific assessment factors. So instead of that uncertainty factor, to account for species differences in kinetics, you can use these models to actually show the difference in kinetics between different species. Of course, you can extrapolate from chemical-rich toxicity from data-rich chemicals to toxicity of less data-rich chemicals using these PBK models. You can use them for read-across, for example. You can establish therapeutic doses when it comes to drugs, and that's where this type of modeling actually originates in predominantly. But you can also interpret biomonitoring data. But what we want to use it for really is for QIVIV, quantitative in vitro to in vivo extrapolation. And just bear with me, but the way that you use it is in a reverse dosimetry setting. So instead of giving it an oral dose, here you're using your in vitro concentration response relationship. You put that concentration equal to the concentration in the tissue where that toxicity is relevant. So in this case, we've got glycol ethers and it's alkoxy acetic acid. So these metabolites are very embryotoxic. So we set these metabolite concentrations equal to the concentration in blood, and then literally you back extrapolate what the oral dose should be to reach concentrations in the blood that are associated with toxicity in these in vitro assays. And this is an example, one of the first examples where this is done by Jochen Louise back in 2010. He tested these glycol ethers, or at least the metabolites of the glycol ethers in this mouse embryonic stem cell test. And then you use this PBK model to back extrapolate what the dose of the mother should be, of the mother rat should be, to explain the embryotoxicity of these glycol ethers. And this is for one of them, but the others look equally beautiful. And this is two types of modeling approaches, but you can see that the dose response curve that is simulated in this case actually perfectly overlaps with the points here that were actually seen in an embryo in a developmental toxicity in a developmental toxicity assay. So now remember ToxCast here, and this is one of the last slides. ToxCast, where we had all these chemicals that were tested in a whole plethora of in vitro assays where they all gave a range of effect concentrations, but it didn't really help us, figure out which of the compounds is are most problematic. Once they used this IVIV approach, a QIVIV approach, they translated these effect concentrations into oral doses, in this case, milligrams per kilogram per day. And then you can see here, then there is a significant potency difference between these different compounds, and you can then compare them to the estimated exposures, oral exposures that are associated with the compound. And from there we can see, for example, that triclosan might be a compound that we are being exposed to at levels that cause significant toxicity in these in vitro assays. So that brings me to my conclusions. What's wrong? And I've hopefully answered the questions that I set in the beginning of this presentation. What's wrong with the way that we perform toxicological risk assessments now? Well, the relevance of animal tests for assessing human toxicological risk is questionable. There are issues there. And why are in vitro assays so special in this case? Well, they can provide us some of the mechanistic understanding of toxicity, but they are useless, arguably, if we don't translate these concentrations into doses. So the toxic potency is obviously a function of both the toxicodynamics, which these in vitro assays could help elucidate, but also toxicokinetics. And so this is why we need PBK models, because PBK models helped us translate in vitro effect concentrations to bioequivalent doses in humans, which we then call quantitative in vitro to in vivo extrapolation. And we could use these extrapolated dose response curves then to set our point of departure for risk assessment. And that's it. Thank you very much for listening, and hopefully it was easy enough to follow.
Thanks a lot, Nynke. There is one question in the chat, or two even. The first one is, I have one more question. I'm curious about your thoughts on the limitations of PBK modeling. Particularly, I'd like to know your views on its application in organ-on-a-chip research. Are you also conducting research on OOC?
Yes, actually. So I would argue that that's research in its infancy. But yeah, so we have ourselves, but I know of others that are looking at organs on a chip a lot. And the neat thing about organ on a chip is, in theory, you know, we can you know, simulate, we can understand some of the kinetic processes that a compound can undergo in a single chip in that sense. So they could actually be used almost as a validation for some of these PBK models, or the other way around. But it's integration. So the integration of both PBK modeling and these organs on a chip, I don't know of any studies yet that have attempted this, but I do know of some EU projects where we do.
The potentials are enormous, isn't it?
Yes.
Yeah.
And it also links back to the question that was previously asked also. So it's like, how do we catch the processes that we don't know, you know, very specific processes? And this is, I think, where organs on a chip might be able to help us identify, identify these very specific processes that we need to then include in PBK models.
The other question was raised by Francesco. He said, excellent talk, Nynke. Now, that's what's in, right? Would you consider the potential implementation of the intestinal microbiota in such in vitro models and PBBK modeling, also in the view of the Q-Viva approach?
Yeah, so I really like the question. So yeah, that's an excellent question. Likewise, so there are actually a number of models, including models that we've or studies that we've done in Wageningen University ourselves, where we've integrated the microbiome, and particularly from the perspective of metabolism, right? So extrahepatic metabolism, that dictates why, usually in our case, why the compounds are not being absorbed as much as we think they are absorbed, or where we've looked at mycotoxins that where the microbiota create an endocrine disrupting metabolite that is then absorbed to cause endocrine disruption.
Mm-hmm.
The integration of microbiome, intestinal microbiome studies and PBK models is actually a field where a number of us work in.
Okay, thanks. Well, thank you very much for your presentation. And it's now time for first place, I would like to thank Mathieu as well as Nynke for their excellent presentations. One remark I would like to make is that, of course, they are very overwhelming, both of them. Be assured that the meaning of the essence of this webinar is to give you a flavor of what's going on. If you want to apply it yourselves, be aware of the fact that one of the reasons that we put it in is that you need to know that this knowledge is there and that you will be able to interpret the results of it rather than do it all on your own. That's, I think, a remark to be made in the beginning. But let's go to the why of this webinar in the beginning. Just to remind you that we started with, let's say, the classical way of risk assessment and why we wanted to change it, and remind you how the present risk assessment is going on. We talked about this very seminal report, toxicity testing in the 21st century from the Americans, as a basis of this. And this also gives you the idea of the systems biology approaches that are very prominent in this report. As a basis of this, we had an expert group consisting of these persons in 2015, 2016. And they came up with the task to see how we could have these new approaches integrated in the way in which we will be able to do this in the novel foods, food ingredients area. We talked about new technologies, new concepts, and especially relevant for the human consumer. That was the starting point. The scope was, let's see what is going on. Can we put it also in the field of mixtures? what are the gaps lower than the no effect level or the benchmark dose or whatever you would like to have it. So just an example on what could be done. We also talked about the choice of in vitro bioassays. If you know more about the structure of the compound, and if you know more about the exposure, and if you are will also be able to include, let's say, the kinetics of the compound, then you will be able to select the proper in vitro bioassays on the basis of the fact that the kinetics predicts that the concentration for a certain exposure will be high in certain tissues. So focus on these tissues in vitro. We also talked about the possibility to high throughput systems to be used, etc., etc. So this is one of the boxes in the scheme that can be evaluated in this way. Another issue is, can we activate certain adverse pathways? Do we know something about the possibility that a chemical will be able to enter into certain pathways? For instance, is it a reactive oxygen species producing compound, etc., etc. All these things can be done. And finally, of course, we have to talk about how can we come up with the possibility to use it in a reference dose for reference daily intake, for instance, or AD acceptable daily intake things. And that was the story of Nynke, where we came up with the possibility to do PBPK modeling and qui vive, and that would then lead to safety estimates, for instance, a relevant a reference dose for the risk assessment. So we also said in this this paper, we need showcases. We need to show that it works, and the successes of these schemes should be shown. And if we don't have a success, where are the problems? How can we improve on the scheme? How can we improve on the use of in vitro methodologies, etc.? It will then highlight research priorities, etc. And that is one of the things that we would like to do in the hands-on training that we now plan to do in July in Brussels in the ILSI offices. We would like to have a number of selected compounds or products, and then go step by step through the scheme and decide if a safety or risk assessment can be made, and if not, what is necessary to do to research to reach it. What are the research priorities to do that? And what we hope to do is that we can come up with a number of possibilities and publish the results of this. So what we aim at is that the participants in this workshop that we will be held in Brussels will also be able to be author of the publications out of this. I see that there are a number of questions or remarks in the chat. So I stop the presentation now and go to the chat.
Bas, maybe I can add something to your last point about the preparations for the training. It might also be good if you have data, own data that you want to use, that you bring it, because the more data we have, the better you can perform such a flow diagram and see what is missing. So that would be very helpful, and that can of course be public data or data produced by yourself or your company. But bring your own data is helpful for the training.
Yes, okay. I see that a number of the questions are referring to having a certificate for attending this workshop, and that will be done dealt with by the office. So please send a mail to Geraldine to do this. There's a question by Baya. How to distinguish adaptive and toxic responses in general? Well, that is of course always an issue in the toxicology. When do we decide that a certain effect is an adverse effect? And that is, well, there is a lot of, let's say, expert knowledge to do this, of course, and that is a case-by-case issue. Can we define the apical endpoint of an AOP so that that we quantitatively can say this is an adverse effect or not? That is the question most of the time. Okay, if there are more questions.
Yes, Bas, I'm not sure if you still want to share the poll with the participants.
Yeah, we have a number of questions to the participants now. The registration for this workshop in Brussels is still open. We need to have a minimum participation there, otherwise it doesn't make sense to do it. It's too much work for 1 or 2 people or so, but we have also a maximum. So if the number is low, we have to probably think about another possibility to do this. If the number is too high, we probably have to repeat the whole exercise on another moment. So between 5 and 15 is our goal, more or less, to this. So if we can have the poll and you can express your appreciation of this and also express your interest in where you want to go to. So these are the questions. I see that there is plenty of interest in doing these courses, and there are now 4 possible people that say that we would like to do it in July. Well, this is the first indication, of course. Some people are saying that they cannot do it in July, but on another moment. We give them 16 out of 25 persons that are still in the call have answered the question. So, Geraldine, when do we close the, what is the deadline for subscription?
The deadline is the 7th of June.
The 7th of June, okay.
Yes.
So if you're interested, please contact Geraldine or read the information on the ILSI website for the registration.
I shared the link for registration on the chat. On the Zoom chat.
Okay, so you can find it there.
2 minutes of poll. Maybe I'm giving more 10 seconds in case someone wants to vote. Otherwise, I will share the results.
Yeah. So 10, 9, 8, 7.
I think, yeah. I think here we go.
We will close it. Yeah.
Yeah. Thank you all. Here we go. That's the results of the poll.
Okay. Right. Thank you very much for this. We now have 4 participants that would like to join us. Maybe there is 1 or 2 that would come more. Yeah, I understand that if you're from India, it's very difficult to attend. But, well, maybe we can do it on another way then. At this moment, I would like to thank all the speakers in this course, Cyril, Nynke, Mathieu, for their presentations. I would like to thank Geraldine and Hugo for the support from the ILSI office. And I would, most of all, I would like to thank all the participants, because it was a pleasure to do these presentations for you. And by this, I would like to stop to finalize this, and hopefully see you in July in Brussels. Thank you very much indeed.